So Sam Baker is the CEO of WriggleBrew. WriggleBrew is a startup in Central Florida that's dedicated to sustainable agriculture solutions. With a background in economics and research experience across multiple scientific disciplines, Sam has spearheaded WriggleBrew's growth, including the launch of its flagship organic plant growth accelerator. A lifelong innovator, he has earned honors like the Cade Prize and recognition as an Inno Under 25 honoree. He's an Eagle Scout, a scuba diver, and an environmental advocate. He continues to drive WriggleBrew's mission to improve agriculture and protect ecosystems. So, Sam, we're super lucky to have you here, and I know I'm super excited to hear you talk about worms and plastic. So, with that, I'm going to stop sharing, and you can go ahead and start.
Thank you very much. I really appreciate the introduction. So it's good to see so many people here. I'm going to be talking about earthworms and about some other worms and how they are useful not only for agricultural purposes, but also we have discovered these worms are useful for eating and digesting plastic waste. So I'm going to share my screen real quick so I can show you guys basically a chemistry and biology lesson, but also kind of an overview of the work that I've done. So let me get started with that. All right. You should be able to see my screen right now. I have on here a picture of a piece of polystyrene plastic.
Yep.
And this is essentially a very common plastic. It's a plastic that we've encountered a lot in our lives. It's something we make food packaging out of. It's something we make housing insulation materials out of. It is a polymer that makes up something about like 25% of all landfill volume in terms of waste volume. It makes up a significant amount of the mass of landfills as well. For example, in my home county of Seminole County here in Florida, about 3 to 5% of the entire waste stream is made up of this plastic. And this plastic and plastics like it contribute to a lot of really negative consequences environmentally. They form microplastics. They are eaten by a lot of organisms and can end up choking them to death. But more recently, it's been discovered that a lot of these plastics have very deleterious chemical and biological impacts on organisms beyond just being a choking hazard. A lot of them have been now related to endocrine problems. So that's the hormone system, the hormone regulation system in our bodies and in the bodies of other animals. These plastics have been found to decompose into relatively harmful compounds, many of which are carcinogenic. So that means they lead to the creation of cancers. And they've been linked to a number of diseases and just about every system of the body they inundate. And even more frightening and kind of unfortunate is that now in every single human that has ever been studied for microplastics, for the presence of these tiny, tiny pieces of plastic waste, every organ ever studied has had these found inside of them. There has not yet been an organ in the human body that has been found free of microplastics. This includes all of our favorite organs like our brain and our reproductive equipment, but it also includes the organs of our unborn children for women when they're pregnant. Microplastics can even be found inside of the baby before it's born. They can be found inside of all of our reproductive organs from the time we are children until we grow up. And we still don't really know all the impacts of these plastics. So plastics are a major problem. But they're not really going away anytime soon because they're extremely useful. In the agricultural industry, plastic is everywhere. It's used in irrigation. It's used in IBC totes that people fill with waters or fertilizers. It's used in the polymerized granular fertilizers that we apply. So there's an enormous amount of plastic in agriculture. There's plastic in packaging that goes into food. So we can't get away from it. And it's very difficult economically to get rid of it. A lot of the recycling efforts around it have failed. So it's this enormous, really challenging issue. Yet, despite how toxic these materials are, and I, just went on a bit of a tirade against them, there are actually organisms that have adapted to eat them as a food source. So this is a worm doing just that. This is actually a larva of a darkling beetle. This is Zophobos morio, and they, in this photo, are chewing on a piece of styrofoam, of polystyrene plastic, the plastic I first introduced at the beginning of this talk. And they are using it as a food source, which means they are not just chewing on this plastic in the same way that a toddler does or a kid does when you give them like a toy or something. They are actually shredding it up, swallowing it and digesting it. And it's breaking apart chemically in their stomach and being used for energy. And it's contributing to their biomass. So they're able to subsist on this. And this is a relatively recent discovery that worms are able to do this. It's not limited, I should note, to these worms. There are actually several worms now that have been observed eating these plastics. Both larvae, like these Zophobos morio, Tenebrio molitar, a cousin of them as well, but also black soldier fly larvae have been observed doing this. And even earthworms have been observed doing this, though to a very minor degree. So several organisms seemingly have adapted this method to eat this material. And it raises several very interesting questions. And those questions became the heart of the exploration myself and a few of my friends embarked on to try and understand this phenomenon. Those questions that were raised among us are, well, if we can feed worms plastic, primarily, why aren't we doing that? But how much plastic can they eat? What is happening to the plastic they digest? How does their feces change when it's actually being produced from primarily a plastic diet? Um, can that feces be used for anything? Farmers and agriculturalists are typically aware that manure, for example, is a really good source of nutrients. So can plastic manure be useful? And a host of other questions. And so I'm going to take you through kind of how we answered those questions tonight, and also how the answers to those questions might be useful to you or to anyone you might know, and to generally the future of agriculture. So before I get too much more into this, I'm going to give you a little bit of a brief introduction about me and my business so you know why I care about this sort of thing. Um, if you were at the previous talk, this will be condensed, so hopefully it won't be too much of an imposition. But let me begin with my introduction of my business. So my business is called WriggleBrew, and we are an earthworm technology business. We are based around the idea of reinventing organic fertilizer, or at least a portion of organic fertilizer, by utilizing earthworm manure, which is a more commonly known as worm castings, as a feedstock for some advanced fertilizer. We're a team of people that met in high school and in college, and we've stuck together because we have a very strong interest in science, chemistry, and biology. And we are extraordinarily interested in fertilizer because even though we were chemistry and biology and biochem students, we live in Florida, and Florida is a state that is very strongly affected by fertilizer runoff, particularly from the phosphorus and nitrogen fertilizers. And those fertilizers lead to red tide. They lead to enormous algal blooms, so large that you can actually see them from space. And so myself and my best friend Gabe decided that we wanted to create an alternative to those inorganic fertilizers that retained their effectiveness and retained their value add to the farmer. We found out that earthworm castings were a really, really good way to create an alternative fertilizer. Plenty of farmers found that worm teas were useful, but we realized they could be created, or rather, they could be modified to be more useful. Your traditional worm tea has a shelf life of something like 24 to 48 hours. We developed a method to control the microbiology inside the worm tea so that its shelf life is not 24 hours, but is instead 24 months or longer. And that allowed us to produce something that could actually be shipped to farmers. We found a method to standardize the actual production of the fertilizer. We found standardized diets to feed worms that at most efficiently encourage them to produce compounds useful for plants. And we've been selling this fertilizer now for 2 years to farmers growing things like soy and corn and other crops. And we are now able to effectively offset a lot of the nitrogen and phosphorus fertilizer that would have instead have been used instead of our stuff. And that contributes to reducing things like red tide in Florida, but also to soil degradation around the country. In the process of conducting our research into earthworms for fertilizer use, we stumbled upon this phenomenon of worms eating plastic. So this was first reported in around 2017 or so. And it was found that waxworms and mealworms were the first to be found that could do this, were able to eat polystyrene plastic foam when they were given nothing else to eat. When you kind of trap them in a chamber with it, they'll actually eat it and break it down. And while we were doing our background research for using earthworms for fertilizer production, we learned about this and became very fascinated and interested in the possibility of using plastic digestate, so the digested plastic material that these worms are eating, as an additional source for fertilizer. Because if someone could take this, 5 billion pound a year problem where we're dumping things like styrofoam and other plastics into landfills and convert that through some special process into earthworm manure, worm castings, then they could not only create fertilizer like the fertilizer we're creating, but they could also create other varieties of fertilizer. They could create earthworm castings that could be used in agriculture or could simply be used to dispose of the plastic waste in bulk. So today we're making a really good fertilizer out of this. We haven't yet introduced the plastic digestion to our fertilizer production process, but we have actually begun the process of growing plants and testing plants with fertilizer produced from this plastic digestion process. So I'm going to return to this presentation and start discussing why worms are able to eat plastic. So these guys here, these Tenebrium molitor, they are eating polystyrene. If you remember from chemistry class, polymers, like polystyrene, are really just chains of a certain molecule, a monomer, that are linked together in a very long line. And in the case of polystyrene, the plastic we call styrofoam, that consists really of the styrene monomer, the styrene molecule, linked together in a really, really long chain. And the styrene monomer, if we look at it just from a visual perspective, is actually very similar to another molecule that worms come across a lot in their digestive process. And this is toluene. So toluene is this molecule here. It's extremely similar to styrene. And toluene is something that worms have actually been able to eat for millions of years because it's a compound that we naturally find in things like pine resin. It's a compound that shows up in the detritus of tree bark. So worms encounter this compound a lot. And as a consequence, they have had this evolutionary pressure on them from high amounts of styrene in the soil that has enabled them to adapt to digesting it and using it as a food source. And there is actually an enzymatic process that is produced by the microbes inside of these worms, specifically keyed for breaking down toluene. And it turns out, because of the chemical similarities between toluene and styrene, if you put worms in an environment full of polystyrene, which is just styrene all linked together in a big foam, they will eventually, when they have nothing else to eat, start chewing into it. And the microbes in their gut, which have adapted over millions of years to digest toluene, will start functioning on styrene, albeit it's not extremely efficient, and the worms are only going to go about doing this really when they're starving, but they are actually going to be able to effectively digest this. And styrene itself is not too dissimilar to a lot of other plastics out there, so this is a very encouraging thing. So this phenomenon is very interesting, and there are a lot of studies that have found this, but incidentally, or not incidentally, but interestingly, there are no companies that are really based on this idea. There are no companies that are using this digestive pathway to dispose of this massive plastic problem. So myself and my best friend Gabe, we had already started our earthworm business. We had won at this point $125,000 of funding from the USDA, and we'd been able to make some serious headway with our research into producing earthworm fertilizers. We decided that we would take up this task and see if we could make it possible for worms to commercially digest plastic. And so we decided to propose a series of experiments, mostly to the National Science Foundation, to see if they would be willing to fund us to try and take this digestion mechanism that has been observed and use it to accelerate the process of polystyrene decomposition. See if we could maybe modify the digestive work of these worms so that they could eat this plastic a little more efficiently. And that research began with Worm City. So this is Worm City here. It is a tower full of little chambers with shredded up polystyrene in it and a bunch of different kinds of worms. So in some rows of this tower, we have larvae, we have waxworms, we have mealworms, we have superworms. We also have earthworms, we have black soldier fly larvae, and they are all engaged in trying to eat the most amount of polystyrene in the fastest amount of time. So we found that when we reviewed the literature, there weren't really any studies that gave a one-to-one comparison of what different species did and how well they did. and the digestion of polystyrene. There were no standardized experiments that studied what conditions optimized for the digestion of polystyrene. So our first step was just to kind of replicate the existing literature and see if we could get this polystyrene digestion to happen as it was reported in literatures, and then get some numbers to compare. And what we found is that a lot of what was reported in literature was pretty wildly inaccurate. So there were a lot of studies published on the digestion rates, and they were pretty much all wrong. Many of them reported that the worms would only be able to eat milligrams of polystyrene a year. That was completely wrong. We found that, in fact, they will eat several grams in a few months. These are superworms. They proved to be the most effective of all species, Zoophobos morio. They were able to eat about 10 grams of shredded polystyrene in about 6 weeks. So that's a lot more plastic and a lot more quickly than was originally published or was originally reported. We also found that the earthworms will not do a very good job themselves eating the polystyrene plastic, but they will do a good job eating the feces of worms that do eat the polystyrene plastic. But all this raises a lot of interesting questions because, again, it's showing that, all right, this is still a relatively new field. The literature is still not very accurate. Perhaps if we simply change the conditions of the chamber more, we can get the worms to eat even more plastic. 10 grams in 6 weeks is better than what we found in literature, but it's still, it's still not amazing. So perhaps we could do something like scale up the worm chamber. So you'll notice in this chamber, there's only really about 100 worms. And that's because you can't fit that many in a chamber, but it's also because we want to have lots of different replications with lots of different chambers. In science, you need to have typically 32 independent quanta in your experiment, or not independent quanta, but 32 different replications in your experiment. If you're trying to show whether or not a thing you do has a statistically significant impact on the results, you need to replicate it at least 32 times. So in this case, we have 32 bends so that we can say, okay, changing one variable at a time, let's say the diameter to which we shred the plastic, we know whether or not that actually makes an impact. We found in this experiment that shredding the plastic to different sizes didn't really make a huge impact. The worms were kind of able to eat it no matter what. But we weren't able to test the actual chamber size. So the question was, what if we increase the chamber size? Because with worms, there's kind of a unique mechanism they have for digestion. When they live together communally, like they do in this chamber, the worm, and this is my horrible drawing of a worm with his little mouth and his orifice, as he eats material, as it goes into him, that material is digested by microbes inside of his gut. And when it comes out, yes, it's feces, but it also carries with it some of the microbes, and this is my drawing of a microbe, some of the microbes from inside his gut. So in theory, if you breed a bunch of worms together in a large chamber, then those worms, and I'm going to use a polygon this time, then those worms should essentially output bacteria which are more and more adapted over time to eating the material around them. And because they're outputting that bacteria into the chamber, other worms will start eating material that has these bacteria coating those surfaces. So the fecal bacteria of one worm will be transferred orally to another one. And while that is gross, that is actually one of the methods worms use in soil to boost their population and to increase their effectiveness in producing fertilizer material. I should note worms are incentivized to produce fertilizer material, to produce good castings, because good castings in good soil strata means more leaf litter and more detritus for them to eat further down the line. So it stands to reason that if we get a lot of worms together with a lot of plastic, there might be a non-linear relationship. So we might see that as the population of worms increases by a linear factor, we might see that the digestion, so this is worm population, you might see that the digestion rate does, something like this, let's say. Maybe it's a curve, maybe it's logarithmic, maybe it does something like this. But inevitably, we're hoping that larger population of worms means a larger digestion rate. So, does that work? No. It does work a little bit. So what we find is that when you have lots of worms together, they do start passing around a lot of the bacteria to digest plastics, but plastics themselves are not a fantastic source of nutrients. So the worms are kind of malnourished, which means they're not very healthy to begin with. So pathogens passing around between them hit them a lot harder than they normally would. So when you scale up the worm chamber, you do get better digestion sometimes, but you also get a lot of worm death, and that is really problematic, which is annoying. And the other problem is the worms are making a mess. So, as you can see in this picture, as they eat, they're kind of shredding the plastic up. Well, that means that when they digest a lot of it, they're leaving a whole bunch of little pieces of plastic everywhere. So, we can't just scale up the worm chamber and mix it in with some food to keep their health up, because inevitably that will create a bunch of microplastics essentially mixed in with your food, which is kind of not really solving the problem you wanted to solve in the first place. So that sucks. But what if we go the opposite direction with it? So, what if instead of feeding the worms large chunks of plastic, we scale the plastic down in size? So, what if we take the foam of the plastic, which is a material that is mostly air? Styrofoam plastic is something like 95% air by volume, and we densify it. Well, the reason we want to densify it is because the worms, as I mentioned, they're chewing into this and they're digesting it. Well, a lot of the digestion work is really happening because it's all really happening because the microbes in their gut. So if we can make the plastic denser, maybe every bite they take, they'll get a lot more plastic out of it, and then a lot more plastic will be digested a lot more quickly. So, we try and shrink it down and see if that just makes the whole process faster. And there's actually several different ways to shrink the plastic down. You can do it thermally. You can heat it up and kind of compress it. Or you can use solvents. So, for example, acetone or gasoline, as a lot of kids learn when they're doing pyrotechnics as teenagers, are fantastic compounds because they break apart some of the longer chains of the polymer and they cause the air bubbles or the air to bubble out of the polymer material. And we found that when we used acetone, for example, you get a shrinkage that is very, very considerable. So you get a material that takes up, let's say 2 gallons or so, and it shrinks down to being like 2 ounces. In fact, I've got a picture somewhere with me next to a dumpster's worth of polystyrene plastic before and after. I'll pull that up for you really quick. It's kind of a fun picture. But what happens when we try that? Well, if we shrink the plastic down, we get it to a really small size. We find that, yes, you do get the worms eating more plastic per bite, but you also find that the worms are much more likely to choke and die on the material because denser plastic means much harder to chew plastic, and the worms are still trying to chew it. And so, as a consequence, that doesn't really work either, which is really, really unfortunate. But both of these teach us some very important lessons about how the worms function, and also kind of reveal the entire problem with worms eating plastic in the first place. And that is that the worms are kind of prima donnas. So I'm a huge worm advocate. I advocate for earthworms all the time and how useful they are for our agriculture. But when it comes to eating toxic materials, there's only so much they can handle, and they can only do it so fast. And the worms, in this case, are not really able to eat all that much plastic material because they're choking on it, they're getting killed by disease, they're having all these other problems. And it makes us reflect on why the worms are even able to do this in the first place. The worms are only able to eat the plastic, not because worms are magical and have some special ability over other organisms, but it's because they just so happen to have developed the right microbiology in their gut because they've been exposed to these kinds of compounds over the course of millions of years in nature. It's really essentially the microbes that are doing the digestion, not the worm. The worm is really just acting as a vessel to get those microbes to those pieces of plastic. So the question becomes, what if we just take the worm out of the equation? We use the worm to breed microbes that are plastic digestion capable, and then we isolate those microbes from the worm, and we use those microbes to actually eat the plastic directly. So that was the next thing we decided to try. We were able to eventually isolate the bacteria from the gut of the worm that can digest styrene and toluene. and we were able to breed them separate of the worm, which meant we don't have to cut any more worms up to actually get these materials, or rather these bacteria. And it also means that if these bacteria can be bred, can be reproduced by us in glassware, then potentially we could feed plastic material to them in that glassware. So that's what we decided to do. Instead of having the worm, we decided to simulate the worm gut to build a bioreactor, a chamber, a big glass vessel that holds essentially all the different microbes from inside the worm, and emulates the condition of the worm's gut. So the same kind of vegetation, the same kind of temperature, same kind of oxygenation. And into this artificial worm gut, we feed a liquefied form of the plastic. The reason we do this is because feeding it hard chunks of polymer of the actual plastic would take the microbes a lot more energy to depolymerize. Remember, the worms are actually doing a lot of the work for the microbes because they're mechanically shredding the plastic and they're exposing it to their digestive juices, which break the polymer down into smaller and smaller pieces. Well, we can artificially do that with a process called pyrolysis. We can use a heated chamber that doesn't have any oxygen in it to take the plastic and to liquefy it. And once it's liquefied, we are able to then drip feed that into our simulated worm gut. And we found that that works really well for encouraging the digestion of the plastic material. So we found that in a mason jar bioreactor, which is a very simple bioreactor that you can produce when you are fresh out of university, about 100 grams of plastic material can be digested by microbes in about a quart of liquid in 5 days. And that is a really significant milestone because remember earlier I mentioned the fastest we were able to get it with the worms was about 10 grams of digestion in about 6 weeks. And that includes with them shredding it up and leaving residue everywhere. In this case, there's no residue, and it's being digested to the tune of 100 grams. So that's a 10 times increase in the actual quantity of plastic digested, and in 5 weeks or 5 days rather than 6 weeks. So that's a many times increase in the speediness of it, almost like a 6 times or 7 or 8 times increase. So that's really, really good. So you're seeing a huge amount of plastic digestion. But what about plastic residue? I mentioned the worms, when they chew it up, they're leaving little bits of residue everywhere. Well, when the microbes are digesting it, they're digesting this liquidized plastic we're producing. We found no trace of the plastic. It is being digested 100%, in at least down to the parts per million and parts per billion. So there's no plastic left. And that is really, really cool for a couple reasons. But one of the big ones is that it really demonstrates that the worms, or rather the microbes, are actually getting it done. They're actually eating it. And it also demonstrates that the residue not being there means that this is a safe material they're likely digesting it into. And I've actually got a much better picture of it here. But you can see in this picture some successful reactors and some failed reactors. So, and these guys over here, what we can see is the bioreactors where the microbes were not inoculated or were not inoculated correctly. The styrene layer, the layer of plastic, essentially on top of the liquid, remains. Whereas those where they were not inoculated with the microbes, or rather they were inoculated with the microbes, the plastic layer is gone. So that layer is being clearly digested. There's also a color change in the broth because of all the microbial activity that's happening. But the big part is that material is gone. And when we look at the actual detector we used to check whether or not there's any trace of styrene, which is the monomer, indicative of the plastic still being there, there is no detection. So that's really, really good and very, very interesting. And it also means that this material that we're producing, like I said, is safe, but it also means that it's likely biodegradable. Because when worms digest material, what do they turn it into? They turn it into poop, worm poop. Well, that's a mixture of microbial detritus as well as just some bacterial material, but it's also the kinds of stuff that you find in worm poop naturally. So we chemically analyzed this broth and what was in it. And aside from the normal bacterial stuff and the secondary metabolites you'd expect to find, it is chemically indistinguishable from the poop you find inside of a worm eating normal organic material. It's rich in a bunch of uh, uh, polyhydroxyl alkanoates, which is a class of organic acid, and a bunch of other organic acids like phenylacetic acid. But these are all compounds you expect to find in normal worm poop. So what that means is the digestion is a complete destruction of the plastic material. But it also means that this plastic that's been digested is being turned into something that we could potentially use as a nutrient source for plants. In fact, we jellified it. So we mixed some agar with it and made worm poop jello. And this worm poop jello is a material that biodegrades very rapidly. In fact, we found mold growing on it really quickly, like within a day. And when we put it in soil, it breaks apart into the soil with no residues. There's no plastic that you can find in it, and no toxic compounds. But it is also a nutrient source, meaning you can take this and it has these things that break down into sugars and other compounds, and you can feed that to all kinds of decomposer organisms, or you can mix it into soil directly. We found that if it's fed to earthworms, the earthworms will actually eat that material and poop out basically worm castings. Literally no chemical difference that we could find in the worm castings produced with this digested plastic material and the worm castings produced with normal food stuffs like coconut coir and things like that. So that's really good. That's really interesting. But the natural question is, what happens if you grow plants in it? And the answer is nothing, nothing different than what you'd expect from worm castings. The plants grown in the worm castings derived from the digested plastic grow the same as the plants grown in worm castings derived from ordinary sources. And both do a heck of a lot better than plants grown in just regular, coconut coir or rock wool or whatever. And so what we're seeing here is that the digested plastic is being transformed into a material that actually has value, that can actually be used as a fertilizer. And that has serious chemical, biological, and economic implications that are really, really fascinating. Because at present, when we take things like polystyrene plastic, the 99% of it's just thrown away. It's just thrown into the garbage. Because even if you recycle it, its recycling value is like 10 cents per pound at best. And that's if it's cleaned, if it's processed, and everything. And 90% of the time, it's not. So 10 cents per pound is the best you can possibly hope for if you turn this plastic waste into more plastic, which will inevitably end up being thrown away anyway. So it's not economically viable to recycle it, which is why recycling has never been very successful. But if you transform this into earthworm castings, well, earthworm castings have a market price of between 30 to 40, even 60 cents a pound bulk when you're selling them by the super sack. When you brew them into liquids, then that goes even further in terms of the economic activity you can generate. Or when you use them as mixtures into other soils, you can get up to $1, $2 a pound. And when you sell them as a retail product in a bag of soil at a store, I mean that then you're selling it for like $9 for a 5-pound bag or something like that. That is exponentially more valuable than trying to sell it as regular plastic material. And that means the process can pay for itself. So you can collect the plastic waste and you can turn it into something valuable, which justifies all the extra processing aside from simply shipping it to a landfill. And it gets even more interesting because recall how I mentioned that while the worms are digesting this material, they're turning it into poop, into biodegradable material. Well, what's allowing them to do that are the same enzymes that are being used originally for toluene digestion. And those enzymes are a shelf-stable compound that could, in theory, be mass-produced and harvested from the bioreactor, the chamber where you're growing the microbes in contact with the plastic, which means you can digest plastic, and at the end of the digestion cycle, you will have a tank full of enzymes, which could be freeze-dried and potentially shipped to farms or to landfills or to other places that have high concentrations of microplastics in their soil, and it could then be used to break those microplastics apart in situ, in the field. You dump the enzyme over the field, and it wipes out tiny particles of microplastic in that soil. And so that is an extremely exciting possibility. And so what we've now done is we've begun the process of breeding these microbes more so that we can try and optimize their plastic digestion process, because I mentioned these are microbes that adapted over millions of years to eat things like toluene. But in theory, we could simply speed up their evolution in our biochamber by continuously exposing them to plastics, and thereby hopefully make them even better at digesting just this material. We could also potentially expose them to other kinds of plastic. So right now we're having them eat polystyrene, but there are things like high density polyethylene, which is what makes up, if you're watching this from a computer, the plastic on your computer, whether it's on your monitor or whether it's on your laptop body or whatever, is made of high density polyethylene. So are cell phone cases and a whole bunch of other things like that. If you're drinking milk in the morning, well, the milk jug is made of high density polyethylene. Those are plastics that break down into something called benzoic acid if they are pyrolyzed. And benzoic acid is very similar to toluene and to styrene, which means potentially that could be digested by these microbes as well. And so we were able to successfully pitch this to the National Science Foundation. They had a board of PhDs review our research, and they liked it so much they funded us $275,000 to turn this into a real business. And we've completed this round of research, and we've now requested an additional $1.2 million from the National Science Foundation to turn this into an actual large-scale commercial endeavor where we can be collecting plastic and converting it into soil. And I'm happy to report that as of about like a week ago, we have converted at this point about 400 pounds of plastic so far, which is a really significant amount. And it's a really amazing achievement, I think, because it's one of the first times where plastic is actually being destroyed for good and turned into soil. And that soil we've already given away to a number of farmers who have seen some pretty positive results. One of the interesting ones being that apparently the worm casting soil produced by this method seems to have some really good properties for holding in moisture. But I don't have too much data to share on that yet, so I won't get ahead of myself there. But that is my presentation, and I wanted to open the floor to any questions if anyone has anything they'd like to ask me about this. And I'm very grateful for all your time.
Thank you so much, Sam.
You're welcome.
This is really amazing. Yeah, it's gonna need to
I just
marinate.
can't believe we always thought plastic was something that, yeah, just like it's constantly being recycled, but there's no way to truly get rid of it. This is the
No, there
first
isn't.
thing I've ever heard where you actually are getting rid of it. And then time, like another benefit is creating nutrient-rich soil. So awesome.
That's right. Here's actually the picture I was going to show earlier. I finally found it of the the dumpster worth of polystyrene being compressed into like less than half of a 5-gallon bucket full. And that's from the acetone just using to shrink it down. After that, we can then take it and pyrolyze it and then feed this to the microbes. Uh, but it's a really good thing because it's getting rid of stuff that would fill up space in a landfill. Uh, but yeah, if anyone has any questions, feel free to put them in the chat or put your hand up. I'd be more than happy to talk about this process and worms in general and how they can be used to fight plastic.
Do you think you might, well, I guess you would have to see more data from farmers, but do you have any plans to add the digested plastic to your WriggleBrew formula?
Yes, we do. We haven't done it yet. We're still building up data. We want to have a lot of third-party testing, so that we can say, okay, for a fact, there is no plastic in here. We, as a third party with no affiliation to WriggleBrew, have looked into it and we've grown plants with it and we've put those plants through trials and everything, and it all looks good. But at least for now, this is a process where we're able to get rid of the plastic and just turn it into at least a biodegradable material.
Sam, you
Mhm.
have an interested investor.
Oh, cool. Well, if you're interested, you can reach out to me, and I'm more than happy to talk about WriggleBrew and all of our business stuff. You can reach me at sam@wrigglebrew.com. I will put this in the chat. And yeah, feel free to send me an email and I'd be happy to talk.
And we can send Sam's contact out in the follow-up as well for anyone that wants to reach out. Okay,
Certainly.
Greg has a question. What does your testing show is the inoculation rate for use on typical product like compost tea for foliar spray or extract, so soil or seed coat prior to plant? Are you selling this yet?
Mhm. This is a very interesting question. So we typically do about 2 to 4 ounces for the typical WriggleBrew dilution rate. So 2 to 4 ounces to a gallon of water. So you'll take 2 ounces of it, which is, it's like a quarter of a cup or so, and you mix that in with a gallon of water, and you can spray that like a compost tea as a foliar, or you can pour that on the soil near the roots. So worm teas and compost teas are really good near root zones because they're full of a lot of things like humic acid, which are really good for root health, but they're also very rich in microbes. So in our case, we have trichoderma, different kinds of mycorrhiza and bacillus, and those are really, really best used near root systems because they're going to help with fixing things like phosphorus. They're going to help with nitrogen. They're going to help with some of the health near the roots. They're going to fight some of the agrobacterium, which are bad. So that it's really good to get it near the root system. Um, we use this a lot actually in our testing, kind of as a seed coat. We don't like spray it on seeds, but what we'll do is we will plant a seed, we will put like an eyedropper drop of WriggleBrew near the seed, and then we'll let it go for the rest of the experiment for the next 60 to 90 days or whatever. We found that when we do things like that with worm teas, with our WriggleBrew especially, the germination rate is something like double what if you didn't do that. And the reason being that worm teas and compost teas are often rich in something called gibberellic acid, which is a compound that is very necessary for plants seeds to come out of dormancy. And so it it's something that when you apply like WriggleBrew, for example, you're going to see a lot of more seeds survive to adulthood. And of course, a lot of the other stuff like the humic acid and things are also good for the health of the plant. And yes, to answer the final part of the question, we are selling it. We sell it to quite a lot of farms. So if you're interested, it's available at wrigglebrew.com. We sell the concentrate in sizes everywhere from a 32-ounce bottle all the way up to a 275-gallon tote. So we've got a lot of different sizes for you to try if you're interested. And Wes, I see, has a interesting question. So you said, does each type of plastic beyond polystyrene HDPE need a specific bacteria, or are they capable of multiple types? And that is a question I do not have the answer for yet because we are still trying to figure that out. We believe that it may not be necessary to use more than 2 species to cover the vast majority of plastics. But it isn't known yet whether that will be the case or not. We believe that it will work, but like I said, I don't know because we haven't tested it fully yet. I think that polystyrene, HDPE, LDPE, and polyethylene, which all kind of break down into the same thing and make up the majority of plastic waste, can be disposed of this way. So intuitively, we would expect that this may also apply to the rest. But there are some plastics like PVC, polyvinyl chloride, which has chlorine in it, and that is a very toxic substance. And so that may not do very well when you pyrolyze it and try to feed it to microbes. So that might need a different method. But I think the enzymes produced from this process will certainly work on a lot of the plastics already out there. Randy asks, have you checked what the fungal to bacterial ratio in plastic worm poop is? And no. However, we found, at least just anecdotally from looking at the slides, there's very little fungi. The worms do not tend to carry very many fungi in them. If they do, it's spores usually. They tend to carry bacteria. So it's very, very low fungal population in there. There are some fungi that do digest plastic through entirely different mechanisms, but those are also not very well studied yet, and they have some of their own problems. For example, oyster mushrooms were recently found to actually eat several different kinds of plastics. But a lot of the plastic ends up in their biomass as well. So it becomes kind of difficult to sort one from the other. Sue asks, could you use the product in a hydroponic system? The answer is yes, absolutely. In fact, we've used it in a lot of hydroponic testing, and we found some really great results with it. The WriggleBrew itself, we found, I actually have a picture here from my last presentation. This is an aquaponics system. We foliar sprayed it on these Suriname spinach plants, and we found that this wiped out a mealybug infestation and led to a total recovery in these plants. And that was really, really cool to see because in this case, the worm tea, the WriggleBrew, is not even being used really as a fertilizer. It's really kind of being used as like a biostimulant. But yeah, absolutely can be used in hydroponics, either in the liquid or foliated onto plants growing from that kind of system. Uh, Gus, I see you've got a question. Do you add gibberellic acid or any other plant growth regulators to your liquid product, or is it produced by the microorganisms in it? We don't add any. It all comes from worm castings. So earthworms actually preserve gibberellic acid, auxins, and a host of other plant regulators naturally. They produce what are called surfactant enzymes, which are compounds that wrap themselves up around those plant growth regulators and prevents them from being digested. So earthworm castings are naturally rich in those compounds. So we don't add any artificially. However, some of the microbes that we add do actually produce things like auxins naturally. And I believe some also produce gibberellic acid as well. But we don't add it ourselves. We prefer to let nature produce it or to add it for us. Because usually plants kind of know the ratio of those compounds they want. And we just let the previous generation of plants, which is what we're feeding to our worms, figure that out for them. Jeff asks, maybe a way to break down wind generator blades that are getting buried in the western United States. That is an interesting question, because wind turbines, as you mentioned, are a really serious polluting problem, and they're being kind of just like dumped in the desert right now, which is gross and also pretty irresponsible. So wind turbines, if you weren't aware, are made of primarily like fiberglass. They're made out of a lot of plastic. A big portion of that plastic is epoxy resins. Some of those epoxy resins, I imagine, are probably styrene based because styrene is used for producing things like surfboards. That's how my stepdad used to make his surfboards. So there's a good chance I would think that the wind turbines are probably pretty rich in some of those compounds that these microbes could digest. However, if they're made out of other epoxy resins, I don't know how that would work. But that is a really interesting thought. I appreciate you bringing that up, Jeff, because I would be really curious to try that. We're still in the process of isolating the enzymes from the worm digestive process. So it may be a while till we can test dumping these enzymes on various kinds of in situ plastic waste, such as wind turbines, but I would really be happy for the chance to try it out. So that's a good idea. Thank you for asking. If anyone else has any other questions in those directions, I'm loving these questions so far. These are real thinkers. Or if not, I can show you more pictures of my worms. Cool. All right. Well, that's pretty much it. I don't know how, I think we only have like 6 minutes left in here. But Camille,
Yeah, do you want to show another couple of photos, Sam, and then we'll put up the events calendar. And if anyone else has questions in the meantime, or you think of something, just throw it in the chat.
Yeah, for sure. Let me see if I can find some cool pictures for you guys that aren't just totally gross worms. Ah, here we go. So this one actually, this shows a more detailed portion of the actual digestion process. So you can see that a lot of it proceeds from the digestion of phenylacetic acid. So when you start with something like styrene or whatever, really a lot of the legwork is just getting it to this phenylacetic acid, and once it's there, it breaks down into a host of other compounds. This is the kind of the digestion process where we go from the biogel, the actual goopified or dried digested plastic to the feces, to the vermicompost, and that is kind of fun because worm castings are always just interesting to make. They don't have any smell, if anyone's wondering. If you're wondering whether or not worm poop has a smell, does not have a smell. And finally, here's a picture of our lab, if you're wondering what it looks like. This is our little facility. We're in Orlando, Florida. We do a lot of our own work ourselves, obviously, but we do work with some third-party labs as well to do our analyses.
Sam,
Yeah.
when you were mentioning the sending the freeze-dried bacteria to farmers, would that just work by they apply the freeze-dried bacteria and then once it's wet, it'll get to work?
Basically, yeah. So the hope would be that we could provide these freeze-dried microbes in like a little cylinder or something, and they would put it into perhaps an IBC tote or something like that. and then they
Mhm.
could spray it over their field. Um,
Gotcha.
Alternatively, this could also apply to the enzymes. So enzymes are a bit more shelf stable than microbes are.
Mm-hmm.
So if we could provide them with, again, like a cylinder of these enzymes, same procedure, they would put it into like a big liquid tank and they would spray that over their farm, and then the enzymes would kind of percolate through the soil and start to break down any plastics that they encounter. And since enzymes are really just organic acids that only interact with the one thing that they're kind of designed to interact with, it would have it'd be basically inert and just more calories if it didn't interact with any plastics.
Yeah.
Cool.
Awesome. Thanks, Sam.
You're
I
welcome. It's been
will
a pleasure.
I'll throw up the events for everybody. Um, there we go. Okay, so our next webinar is going to be on February 18th with Brian Massard. He is a cattle rancher out in Montana. He's going to be talking about some really interesting health measurements that he's been studying, looking at using end-soil algae on his pastures and his hay fields, and then the cows eating that through grazing or through feed. So you can register for that on our website. We will also put the link for that in the follow-up email. We have a info session and office hours on the 12th of February. Come hang out, ask questions. We'll do a short presentation, very informal. You can drop in, drop out. And then we have two upcoming in-person events. We'll be at the Southern Farm Show in North Carolina. That's in Raleigh, first week of February, and at High Plains No-Till Conference in Colorado, also the first week of February. So please reach out to us if you have any questions, you want to chat. We'll give you Sam's contact in the follow-up. And then if any of you need to sign in for the CCA credits, just make sure you either scan the QR code or send us an email or a chat on Zoom. Thanks everybody for being here. And again, Sam, we really appreciate you and our partnership together. Have
Yeah,
a great
Sam,
night, everybody.
You're
Sam, if you
welcome.
have any, when you have updates on if you get that grant, let us know so we can keep all these people updated too.
Yeah, I'm gonna find out in late February, so I'll let you guys know.
Woo! Cool. Good luck.
Thank you. All right. Bye
Bye,
bye.
everyone.
Bye, everybody. Thanks.